Hello, Professor. My name is Abraham Tu, and this is my Psychbuster presentation. Today, I'll be going over two claims. The first claim is babies can only see in black and white, and the second claim is having more and shorter study sessions is better than having longer and fewer ones. The first claim is babies can only see in black and white. The following terms will be defined to help familiarize us with the words researchers use. The first term is the retina. The retina is the layer at the back of the eye that receives light and sends visual information to the brain. Then photoreceptors. Photoreceptors are the cells on the retina that receives light and send visual information to the brain. This leads us to the third term, which is cones. Cones are a type of photoreceptor, and they give us color vision. The fourth term is the foveola, the area of the eye that has the highest density of cone photoreceptors in the retina. The fifth term is neonates. Neonates just refer to a newborn under 4 weeks old. The sixth and final term is trichromatic vision. Trichromatic vision refers to the ability to detect red, green, and blue wavelengths. Being able to see all three wavelengths allow for normal color vision. Below, we will be discussing what the research says. Child Development Perspectives, a peer-reviewed academic journal, informs us, contrary to the common view that infants see only in black and white, even neonates can detect some color. Furthermore, the journal stated above informs us through a literature review done by Tiller in 1998, as cited in Skelton 2022, converging evidence from studies suggests that infants' red-green color mechanisms develops first and the blue-yellow mechanisms develop around 4 to 8 weeks later, with infants being trichromatic by 3 months. This means by 3 months, babies can somewhat see differences in color. In addition, the National Library of Medicine, the world's largest biomedical library, informs us that normally, postnatal development of the human retina involves centripetal migration of the cone photoreceptors into the fovea. The Encyclopedia of Neuroscience also states the foveola contains the highest density of cone photoreceptors in the retina. Cones support color vision. In conclusion, a peer-reviewed academic journal informs us that infants less than 4 weeks old can see some color. This is further supported by another literature review that states that babies can be trichromatic by 3 months. The National Library of Medicine and the Encyclopedia of Neuroscience also informs us that during postnatal development, the foveola becomes densely populated by cones, which helps with color vision. All in all, this evidence suggests that babies can see in color vision. Therefore, the claim babies can only see in black and white is busted. The second claim is having more and shorter study sessions is better than having longer and fewer ones. Let us first go over the terminologies. Distributed practice is when a measurable time lag, 1 second or longer, separates study episodes for a given item. For example, item A appears, item A disappears for some amount of time, and then item A reappears. In contrast, massed practice is when a study episode is not interrupted by any intervening time or items. The third term is the spacing effect, and this refers to enhanced learning, when study episodes for a given item are spaced instead of massed. The fourth and fifth term are intersession interval/gap and retention interval. The intersession interval, or ISI, refers to the interval between each study episode. Going back to the example where item A appears, disappears, and reappears, the ISI refers to the amount of time item A disappeared. The retention interval refers to, or RI, refers to the period from exposure to a particular information to being tested on that particular information. These terms will help us better understand the following findings from the research. According to the University of North Carolina at Chapel Hill's Learning Center website, one of the most impactful learning strategies is distributed practice, spacing out your studying over several short periods of time, over several days and weeks. In addition, according to a meta-analysis paper published in a peer-reviewed journal, part of the Association for Psychological Science, but accessed through the website Augmenting Cognition states, distributing learning across multiple days greatly improves the amount of materials retained for sizable periods of time. These two sources, along with many other studies, show that distributing studying helps retain information better than mass studying. The following two slides are going to focus more on the retention interval, intersession interval, and their effects on learning. Published by the U.S. Department of Education, a research paper part of the Current Directions in Psychological Science, a peer-reviewed journal that covers scientific psychology, did an experiment with learning and recall. In the experiment, In the experiment, participants had to learn the names of some obscure objects. The RI was 6 months, the ISI ranged from 5 minutes to 6 months, and the optimal ISI was roughly 1 month. The 1 month ISI achieved the best recall. The authors concluded that the duration of the gap has a big impact on final recall. Too brief gaps causes poor performance than excessively long gaps. Also published by the US Department of Education, Sapita et al. Did a study where over 1,350 individuals were taught a set of facts. These graphs show accuracy as a function of ISI. The data shows as RI increases, the optimal ISI should increase. The authors concluded that while there are costs to using a gap that is longer than the optimal value, these costs are much smaller than the cost of using too small a gap value, as evidenced by the fact that as gap increases, accuracy increases steeply and then declines much more gradually. Several sources, whether colleges' learning center websites or research papers, show that distributed practice helps with learning. Two research studies published by the US Department of Education demonstrated that the length of ISI is also an important factor in recall. You would rather have a longer than optimum ISI than one that is too short to achieve better recall. Assuming the RI is the same, having more and shorter study sessions would produce a better outcome than longer and fewer ones. This is because the former allows for longer ISIs, which significantly helps with final performance. However, this is assuming that RI is the same. Therefore, this statement is plausible. Below are my references, and thank you so much.